Welcome everyone to the first CIC lecture of the year. I'm Frederick, a third-year PhD candidate in Dr. Sylvia Villeneuve's lab, and I will be the moderator for today. So it is my great pleasure to present today Dr. Bratislav Misic as our speaker. Dr. Misic leads the Network Neuroscience Lab and investigates how cognition, operations, and complex behavior emerge from the connections and interactions among brain regions. The goal of this research is to quantify the effects of disease on brain structure and function. His research program emphasizes representations and models that not only embody the topological organization of the brain, but also capture the complex multi-scale relationships that link brain network topology to dynamic biological processes, such as neural signaling and disease spread. Dr. Misic's research lie at the intersection of network science, dynamical systems, and multivariate statistics with a focus on complex datasets involving multiple neuroimaging modalities, including fMRI, DWI, MEG, EEG, and PET. So it is my great pleasure to have you today. I'm sure the talk will be exciting. Just as a reminder for everyone, as Manal just said, I will be monitoring the chat. So you can raise your hand, write your questions in the chat, and I'll find an opportune moment to interrupt Bratislav with a question when the time's appropriate. So the floor is yours.
Well, so thank you so much, Fred. Thank you, Malar, for the opportunity to speak here. It's a super cool talk series that you guys have, and hopefully over the next 40 or 50 minutes, I'll tell you a little bit about this field of network neuroscience. Obviously, I can't cover everything in the time allotted, but I hope to give you a bird's eye view of what can be done, what's important, and what are some of the really outstanding issues that we think about in the field. So, oh, there we go. You're gonna have to excuse me. I have a very clicky keyboard when I try to change slides. So I'll start out with just building a little bit of intuition about why we would want to represent the brain as a network, what that representation buys us, why it's biologically and useful. I'll spend a very short period of time talking about how we reconstruct brain networks. Obviously, this is a field unto itself, but just so we're kind of on the same page when we talk about these concepts. I'll spend quite a bit of time talking about what are the kind of different features you can derive about brain networks and what are some of the key results that have been reported in the field, what are some of the findings that we all agree about and on. And I hope to spend most of the talk talking about this kind of fourth point here, which is how do we interpret these features and how do we make statistical inferences about them? And finally, I'll show you a few resources that you might want to take a look at if you decide to embark on this in your own work. So just to kind of get us going, all right. So if we think about the system that we're actually trying to study, we're talking about a lot of these intricate units called neurons that are connected with each other via electrochemical synapses. And the function of this complex network that results is really twofold. It's to transmit signals in the form of electrical impulses and also to transport molecules that are needed for growth and repair. Now, we live in an exciting time because we have the means to map, image, and trace the connection patterns in many different types of brains. So we can do this in simple model organisms, something like C. elegans. We can do this in more complex organisms such as the mouse, and most exciting, I think for most of us here in the audience, we can do this also in the living human brain. So what used to be done with painstaking manual dissection and postmortem tissue can now be done relatively automatically. We can use techniques such as diffusion-weighted MRI to reconstruct the white matter fibers that interconnect different neuronal populations to one another. And we can distill these complex data sets down into these wiring diagrams that we call connectomes. And these wiring diagrams simply tell us what is the probability that two neuronal populations are connected with one another, and therefore how they're all integrated into a coherent system. The great thing about representing the brain in this way as a graph or a network of brain regions that are interconnected with one another is that it opens up our field to talk to others in other areas of science. So we can look to our what our colleagues are doing in molecular genetics, to how people represent the relationships between semantic concepts. We can look at the science of sort of transport science. And oh, I think we have a little bit of little background noise. Isil, could you mute yourself, please? Hi, Isil. And finally, we can look to folks who are, for instance, looking at social networks. By representing the brain as a graph, we have access to this lingua franca that allows us to borrow concepts, methods, ways of thinking about how these networks are organized, what's important for determining the roles of individual brain regions, or how they function as a unified integrated system. So the cool thing about the field of network science is that you can actually trace it back almost exactly to this one opinion paper that came out about 15 years ago now. So it's a paper by Alice Porn, Julia Tononi, and Ralph Cotter. And in the paper, they argued that if we wanted to understand the function of the human brain, it would be a really good idea if we had a complete catalog of all the neural elements and the connections between them. And they refer to this object as a quote-unquote connectome, the analogy being with a genome, which is a complete catalog of all the genes and what they do. And this idea has given rise to this very broad field of network neuroscience that concerns itself both with questions that are more technological in nature. So how do we map, record, trace connection patterns in different types of brains, but also more analytical questions such as how do we analyze and model these connections and the interconnection and the interactions that ultimately result from the connection patterns in these systems. The primary methodological framework that we and others have adopted is to represent the brain as a graph. That's what you can see in the bottom set of diagrams here. That is a set of nodes that's interconnected by a set of edges. And in doing so, as I mentioned in the previous slide, we now gain access to kind of a common framework with which to situate our science relative to others, but also share tools and the potential to discover patterns that are universal in nature. To make this a little bit more concrete, really what we get when we represent the brain as a graph is the ability to analyze it in really multiple dimensions. So some of us are asking questions about how the brain is organized across multiple spatial scales. So for instance, how do the relationships that we see among neurons or neuronal populations actually translate to what we see at the macroscopic level? Some of us are interested in how the brain changes over time. So whether that's across very short periods of time, instantaneous, second or minute sort of scale due to experimental manipulations, or how that happens across phylogeny and ontogeny. By representing the brain as a graph, we have a way to capture these changes in a principled manner. And finally, as I'll show you, there exists this kind of third dimension, which is how the brain is organized across multiple topological scales. We can ask questions such as, what are the most important nodes or connections in these systems? What is the, I've lost my cursor. What is the tendency for different units in these systems to cluster together to form modules? And finally, how do they all fit together? And is there anything about the complete organization of the network that supports information processing and functions such as that? So just a very brief overview of how we reconstruct brain networks and what we're really talking about when we do so. So we typically, I'm going to take the perspective of human brain imaging. I understand that maybe some of you are not necessarily in that space, but the concepts are relatively similar. Even if you're working on mouse or macaques or something like this. Basically, we would typically take the brain and we can derive two types of networks. One that's termed structural connectivity. Here we divide the brain up into a set of parcels. So these would become the nodes in our network. And we would use some form of invasive or non-invasive imaging and tracing to reconstruct the white matter fibers that interconnect different parts of the brain. And the end result of all of this is what's called an adjacency matrix. So this is simply a table where you have brain regions in the rows, you have brain regions in the columns, and any one of these elements tells you the probability that two brain areas are connected with one another, that they shared an atomical connection. Now, we can do something very similar, but looking at the activity of these brain areas and using this to construct what are called functional connectivity networks. So here we would, from the same parcels, record either electromagnetic activity or hemodynamic activity, allow us to generate time series. And we would say, if I take two brain areas and I correlate their time courses or look in some other way, look at what is the potential for these areas to co-activate, I can reconstruct an analogous network that I call a functional connectivity network. And here, any one of these elements tells me what is the how sustained are the co-activations between different parts of the brain. The idea here is if two areas display similar fluctuations, then we make the inference that they're engaged in some common function, and maybe they're even exchanging information with one another. But the two types of networks can then be subjected to very similar sets of network analyses. The main thing that I want to impart to you, and I think that's sometimes lost when people apply these graph theoretic measures to brain networks, is that with structural connectivity, really we're talking about the physical infrastructure of the brain that's relatively stable, and it's relatively sparse. So not all areas are connected with one another. With functional connectivity, we're really talking about a statistical concept. We're talking about something that's much more dynamic in nature and that can take on potentially multiple configurations over time. So a lot of the graph measures that you'll see can be applied to both, but some are better suited to one type of network than another, and that's something that you should keep in mind as you do this. Just one slide to show you how we think of the relationship between structure and function. This is from a couple of reviews from the lab, where basically what we think is that the structural connectivity between different parts of the brain provides an infrastructure that supports a myriad of elementary communication processes, and the superposition of these communication processes ultimately manifests as these patterns of interregional covariance that we call functional connectivity. So structural connectivity and functional connectivity are two domains that are empirically accessible to us. We can measure them, we can define them. But the nature of this communication process is something that we try to infer, and something that actually many of these graph measures can help us to do. Now, what do we know about the brain? How do we quantify different aspects of brain structure? Now, this is something that it's very useful to think about this from first principles. So the network representation of a complex system can potentially help us to understand its ultimate function. And if we're talking about very, very small networks, it's very intuitive to do this. So what you're seeing on the left is a toy network. And I can, for instance, ask, what is the most important node in this network? Obviously, one way I can do that is to try to count up the number of connections that every node participates in. And in that case, the red node is obviously the most important one. The blue node is not that important because it's simply not connected to very many other nodes. I can ask, how easy is it for two nodes to send information to one another? And if I wanted to get a sense of this, I might ask, well, if I take two nodes, such as the two red nodes here, what is the minimum number of connections that it would take to get from point A to point B? Obviously, if two nodes can communicate via a very small set of synapses, then they can exchange information more readily. And finally, I can ask, are there any patterns of connections among subgroups of nodes? Is there a tendency for this network to cluster into specialized modules that perhaps are segregated from other modules and perform fundamentally different functions? Obviously, in these types of networks, it's very easy to do. But what about networks where you have many, many more nodes and edges? What if you have something like this? This is the brain of the larval zebrafish. Going through this kind of process of simply looking at the diagram and trying to surmise what are the most influential nodes, what are the functional clusters, how could communication take place on this network, would be very challenging. So what we need is a set of definitions for how we quantify these properties. We need algorithms to do this, and we need a whole science to help us there. Unfortunately, the field of graph theory provides us with a number of features that we can use to quantify and to articulate different aspects of network structure. So I just want to show you one slide about what are some of the key findings. Before we even get to the definitions, what are some of the key findings in the field? And I think it's very instructive to look at this because at a time where a lot of adjacent fields are suffering from a so-called replication crisis, I'd say that in network neuroscience, what we're seeing are very consistent results. Results that seem to appear time and time again across different measurement scales, measurement technologies, different species, and so on. So what you see here is one connectome of the C. elegans, one for Drosophila, and one for the human. What we typically tend to see is a specificity of connection profiles. I'll turn your attention to C. elegans, where the sensory neurons seem to project almost exclusively to interneurons, and the interneurons seem to project almost exclusively to the motor neurons. So there is this kind of the network structure seems to shape the flow of information. But the Drosophila is a really good example because it highlights a lot of these key findings. One is that we tend to see this characteristic combination of high clustering and short path length. So in other words, these networks tend to look like small world networks. What that means is that with high clustering, what we're looking at is how many completed triangles there are in the network. And what that simply tells us is that areas that perform similar functions tend to be densely connected with one another. And at the same time, there exist lots and lots of potentially advantageous routes of communication to go from one part of the network to another. We see a tendency for the network to cluster into network communities. And that's what you're seeing here. So the Drosophila, this is the fruit fly, brain has a very simple structure. It clusters into these 5 modules where you have 2 that transduce visual or photic stimulation, 1 that has to do with taste and olfaction, 1 that's more of an auditory or mechanoreceptive kind of module, and 1 that has to do with orienting the animal and flapping the wings, kind of a premotor module. These areas are all of the kind of neuronal populations within these functional clusters are densely connected with one another, and they share comparably smaller connection density with other parts of the brain. At the same time, there does exist an infrastructure that allows for information to be integrated across this network. And what we see is a broad, heavy-tailed degree distribution with high degree hubs. And what I mean by that is that we see a small number of nodes that are disproportionately well connected. Those nodes are shown to be bigger in this particular diagram, and they tend to be densely connected with each other, above and beyond what you would expect just by chance. So even though we have a tendency for nodes that perform similar functions to cluster together, we still have this infrastructural feature that's superimposed on top of the network that allows for information to be absorbed into these highly connected nodes, but also disseminated to the rest of the network. And like I said, these features are very highly conserved across species, and they seem to be replicable across multiple scales and reconstruction techniques. So let's just spend a little bit of time talking about how we can do this in our own work. When it comes to various graph theoretic measures, there's kind of one way to divide them that makes a little bit of pedagogical sense. So those that are more local in nature, where we're talking about the influence of a particular node or an edge. Those that are concerned with the mesoscale structure of the network, so the tendency for nodes or even edges to cluster into specialized modules. And finally, those that are global, that tell us something about the network as a whole and how it's configured to transmit information. So we'll start with the global measures first. And obviously, because we only have about 40 or 50 minutes, I can't really take you through all the definitions, but I'll try to give you some of the highlights. With global measures, the main advantage is that they will summarize this very complex network structure, typically to just as a single scalar value. The disadvantage is that these measures are not necessarily localized, and one could argue that in some instances they're oversimplifications of the very complex patterns that we get in these networks. So as we go through this, what you'll notice in the bottom of the slides are there are going to be links to the functions that you would actually use to compute the different measures. And the links are from the Brain Connectivity Toolbox. This is a MATLAB toolbox that was developed by Mika Rubinov and Olive Sporn some years ago. It's something that we use in the lab, but there are lots and lots of other toolboxes that you can use if you're more Python-minded. Somebody has made a Python version of this that has the same kind of function names. And then there are a variety of other toolboxes, and we'll go through this as well. But if you are using the MATLAB version of the Brain Connectivity Toolbox, you'll see the links to the functions at the bottom. So when it comes to global measures, the most important, most fundamental of all is the network density. So that's the proportion of possible connections that are actually observed in the network. So here you have both the kind of network representation and the adjacency matrix representation. The density of this graph is simply the number of connections that are present divided by the number of connections that are possible. So in this case, the density of the graph is 0.22. 22% of available connections are actually there. And as I said at the bottom here, you'll see the functions that you would use to do this. If I start adding connections to this graph, you'll see that the density will go up. So you can think of this process going left to right as me either increasing or decreasing the threshold in the graph as to what I'll include. And the most important thing that I want to impart here is that every other graph theoretic measure will scale with density. All graph theoretic measures are relative, and you have to fix the density if you're doing any kind of comparisons. So just to kind of show you that, the path length in this network, so this is the mean shortest set of contiguous edges that it would take you to get from any one part of the network to another, is on the left-hand side, it's 2.62, but as I start adding connections, I now have lots of shortcuts, and the path length will keep decreasing as we add more connections. Conversely, you might be interested in something like clustering. So like I said, this is the number of transitive closures, the number of completed triangles that we have in the network. Here, we don't have any completed triangles, but as we start to add connections, the clustering coefficient increases. So what I'm really just doing is thresholding the graph, but as I change the density, these two fundamental properties are increasing. Sorry? No, thank you. I thought there was a question.
Sorry about that. That was deep opposition to your description, but it seems like
how dare you do clustering? No. And the question then becomes, how do you choose the density in your work? It strongly depends on what you're trying to achieve. One thing that you can do if you're studying just one graph and you're happy with how it's reconstructed is you can keep it as is. Another is to match it to some ground truth. So for instance, if you're doing diffusion imaging, you might want to say that, well, you should not be getting a density that's too different from what we would get from gold standard techniques, such as track tracing. And with track tracing, people report anywhere from 5% to 50% dense connectomes. So you might want to do that. You can set the threshold and therefore the density and vary that statistically. So you can try to demonstrate that the effects that you're getting are immune with respect to how you set your threshold. And the last thing that you can do is if you are working on kind of case control comparisons, or you have multiple conditions, is to match your groups or your conditions for density so that you can actually compare them afterwards. So once you've set the density of your network, now you can get into more interesting questions. Probably the most interesting of these is how information travels on the network. So here it's very useful to make a distinction between what we call walks and what we call paths. With a walk, we're asking what is any kind of non-repeating sequence of edges between two nodes. So if I pick a node, a source node and a target node, how can I kind of make my way from one to another? How easy is it for information to travel on this network? This would be a walk, and the length of this walk would be 5 because there are 5 connections. There could be many such walks, obviously, and this is different from a path, which is the shortest contiguous set of edges between a pair of nodes. So for instance, here I have the same source and target. Now the path is shown as such, and the path length here is 3. The characteristic path length of your network is the mean path length between all pairs of nodes. And if your graph has a very low characteristic path length, or comparably, if it has a very high, what's called global efficiency, then your graph is well organized, well positioned to transmit information between all constituent brain regions. A low path length or a high efficiency would imply that you have a high communication capacity, or that you have a lower potential for signal attenuation, because obviously these impulses are going via electrochemical synapses. They're inherently noisy. The one thing that I just want to mention is that many of these measures that revolve around recovering paths and networks, which people tend to use quite a bit, do make the assumption that the communication that's taking place on the network has some knowledge of the global topology. So for instance, that your neurons actually know what the shortest path is and can compute it. The other assumption here that's being made, of course, is that only a small proportion of the total infrastructure will be used. So this is a brain network that we've reconstructed from diffusion-weighted MRI. It's got 24,000 connections. These are the connections that participate in at least one shortest path in the network. What you can see is as you go from this original network to this kind of shortest path skeleton, you're going from about 24,000 connections to about 6,000 connections. In other words, if you apply these path-based measures, you're really assuming that only a small percentage of your total infrastructure is being used, and the rest is just sort of vestigial. And there exist actually a myriad of other decentralized communication strategies that you could implement on the network that use concepts such as random walks, navigation, and so on. This is something that actually my group studies quite a bit, and then this is just a diagram to show you that there's actually a spectrum of communication strategies that you can apply that are analogous to path-based measures, but potentially biologically more meaningful. So those are some of the global measures that you might be interested in. We're gonna shift our attention over to the other end of the spectrum here, which are the local measures. So this is a radically different way of looking at the brain, where we're now asking what are the influential elements, so we can really pinpoint where our network is different from another network. But the disadvantages that as you'll see, is that many of the measures that have been proposed are sometimes redundant with one another, and oftentimes will make some very specific assumptions about the underlying mechanisms of functional interactions that take place. So when we talk about local measures, I mean, we're asking really how important is a given network element, and there are many different ways to operationalize that question. One is to look at simple, what I would call connectedness, so to count how many connections a node participates in. So that's what's being shown here. We have a graph, and we have a particular node, and we're asking how influential is it. We can estimate what's called a degree, so just how many connections are incident on our node. If a node has many connections, then it's well positioned to influence many of its neighbors. It might be very important in the network. And then we can take this concept and we can really apply it to different ways of reconstructing the network. So those of us that are lucky enough to have connections with directions, so for instance, that are reconstructed from track tracing, can ask what is the in degree of a node versus the out degree of a node. So how many connections are afferent and how many are efferent. And I'll just point out here that you can make actually a lot of very interesting inferences about the role of a node if you look at the ratio of its in degree to its out degree, for instance. We can make guesses as to whether a node is better positioned to absorb information or to disseminate information. We can also, oftentimes, because we have weighted networks, so those of us who are working with diffusion MRI, functional MRI, and so on, we can use weights to gain more insight into the function of these nodes. So for instance, before, the red node was the most important one because it had the highest degree, but if we take our weights into account, we can clearly see that the red node here is participating in a lot of low capacity connections, whereas the node on the left I've now highlighted in red, even though it has fewer connections, it participates in a lot of high bandwidth connections. So its total influence in the network might be greater. And if we count up the total weights of connections that a node participates in, this is what's called the strength of a node. And it illustrates another important concept, which is that almost all of these graph theoretic measures that I'm talking about have not just a binary version, but a weighted version, a signed version, and a directed and undirected version. So those are just some of the ways that we can look at the influence of a node from the perspective of the number of connections that it has, but we can also look at higher order topological effects. So these are, broadly speaking, the statistics that would fall under this umbrella of centrality. So one of the simplest things that people can calculate is the betweenness of a node. If I take a particular node and I reconstruct different shortest paths in a network, I can ask what proportion of shortest paths go through my node of interest. Because if my node sits at the intersection of many different shortest paths in the network, then it's well positioned to act as a bridge or as a way station for signal traffic. It's well positioned to integrate and to disseminate information. We can also ask what is the mean path length from my node to other nodes in the network, which is the so-called closeness centrality. And there are many, many other kind of iterations and versions of this concept. And then finally, there is a class of connections that's used a little bit less often, but I think is also very informative, which is the vulnerability of a node. And here what we're asking is how much would the communication capacity of my network, so for instance, its efficiency or its characteristic path length, change if I was to remove a particular node? Because if the efficiency of my network really drastically goes down after I remove a particular node, then that node is very important for network function. So it's a bit more of an empirical approach to ranking the importance of different network elements. As I said, these measures all have signed analogs, they have edge analogs, and that's an important point. This idea of edge analogs, everything that we've talked about, such as what proportion of paths go through a particular node, I can do the same thing. I can calculate the same thing about an edge. And I can ask, are there particular anatomical projections that are more important than others? And but the one thing just to keep in mind is that many of these measures will be correlated with degree. Obviously, if a node has many connections, then it's always going to be much more central in the network. And as a result, they're often correlated with one another. So what you're seeing on the bottom right-hand corner is a really nice paper from Stuart Oldham in Alex Fornito's group, who went and collected a whole bunch of brain networks and other types of network data sets. And then he computed a wide range of different centrality measures for these networks, and he correlated them with one another. And what you're seeing is that, so that the different measures are in the rows and the columns here. What you can see is that the magnitude of these correlation coefficients is very, very large. In other words, they're all going to be highly correlated with one another. And one thing that I would urge you to do is to always think about whether you really need that very complex statistic, or could you describe the effects that you're interested in with a more simple, straightforward measure. Finally, many of these measures will imply a very specific signaling or transport mechanism. Obviously, the statistics such as betweenness assume that communication is taking place via shortest paths, and there are other assumptions that we'll talk about in a few slides. So those are kind of the two extremes, the local and the global measures. But in between these two extremes, there's actually a rich variety of mesoscale measures that I think are used a little bit less often, but in my opinion, have a lot of power and can give you a lot of important insights into how your network is organized. So here we're talking about the tendency for your network to cluster into groups or patterns. And the advantages are that you can now identify subsystems that are potentially functionally relevant. And the disadvantages, as you will see, is that a lot of these methods are model-based, and there are quite a few moving parts to this, and they're a little bit less straightforward to apply than the local and global measures. So when we talk about mesoscale architecture, the main thing here is that it is rarely visually apparent. So if you have a graph like this, it's not really obvious whether you have any functional clusters. What you would really want to do is reorder your nodes by their membership in these clusters so that you can reveal groups of nodes that have dense connectivity with one another, but relatively few connections with the rest of the brain. So as a result, these modules or communities, as they're sometimes called, must be detected algorithmically. And what I mean by that is that you have to actually define what you mean by a community, not trivial at all. You must specify an algorithm that you're going to do this with, very, very difficult. And finally, these algorithms often have lots and lots of free parameters. So you really have to find a way to optimize the parameters and validate your solution. In the most general sense, community is a group of nodes where the observed density of connections is maximally greater than what you would expect by chance. Concretely, what we mean by that is you have some adjacency matrix. I'm going to denote A, and you have some null model. We'll talk about null models in the last part of the talk. you have some null model that tells you what is the average chance level of connectivity between any pair of areas. And you use that to come up with a kind of an expected connection density matrix. And the difference between the two is sometimes called a modularity matrix, often denoted by the symbol B. And intuitively, a high quality partition into communities is one that's going to concentrate the strongest element of this modularity matrix in the communities. So this would be an example of a high quality partition. You can see a lot of the red elements are in these little blocks along the diagonal. This would be an example of a low quality partition where the red elements are now distributed somewhat evenly across the network, and they're not really that prominent in the communities that we've decided to define. So how do we actually find these? There are many different ways to find communities. In brain imaging, we're obviously very familiar with different types of agglomerative clustering and spectral clustering. People will also use things like ICA. In other fields, simulated annealing is very popular. But really, the way that we do this in network neuroscience is to use what are called community detection algorithms. And there are many, many such algorithms. The most popular of these are those that use a particular quality function that's called a modularity. And so they're called modularity maximization algorithms. And I would say by far the most popular of these modularity maximization algorithms is the so-called Louvain algorithm that's shown here. We can go into the details of how this works if anybody's interested. But the important thing here is that we operationalize our communities as follows. We have this is our quality function, the so-called modularity function that was first introduced by Mark Newman. And the modularity function is just the quality of a given partition of our nodes into communities. The way that it's calculated is very similar to what we saw in the previous slide. We have some observed connection weight between nodes I and J, and we have some expected connection weights between nodes I and J that's shown here, Pij. So this is our null model, Pij. This is the so-called Kronecker delta. So this is just a function that returns 1 when the two arguments are the same and returns 0 when they're different. So what this means, and the two arguments here are the community assignments of node I and node J. So all this is saying is I'm going to go for every pair of areas, and if they fall into the same community, I am going to perform this difference, and I'm going to keep summing these differences. So I'm going to keep looking at how much of a difference is there between the observed and expected connection density in the nodes that fall into my putative communities. And the sum total of this is the so-called modularity. So that final value of Q is really a measure of how prominent or statistically unexpected the within community connections are. Now, the cool thing about modularity maximization is that it is very flexible. And there's just one kind of extension that I want to share that I think is particularly relevant for those of us who are in neuroscience. And that is taking the base kind of modularity function and looking at how it varies across resolutions. So this is just a very simple extension to this formula where I introduce a free parameter that I've called gamma. This is sometimes called the resolution parameter that I'll explain, and I'll explain why in a second. But this parameter simply weighs the importance of my null model. So what that means is that if the parameter is very small, that means that my null model is very weak, and it's very easy to overcome this null model. And as a result, I'm going to push the algorithm to try to find bigger and bigger communities that don't necessarily have super strong density. If, on the other hand, the gamma value is very large, I'm now making the null model much more prominent, and it becomes much harder to overcome. And what this means is that now the algorithm has to really find communities that are very, very densely connected, and then as a result, it's going to try to find a smaller number of a larger number of smaller communities. And the reason this is, I think, very promising for those of us who study the brain is that we believe that the brain is organized as a set of nested and increasingly polyfunctional neural circuits. So being able to parametrically tune how these communities are turning out in our brain networks is very powerful, and gives us a very nice and principled way to describe the community structure of the brain across multiple scales of organization. If you have divided your brain into communities, and I'll say that if you apply this to functional networks, what you're going to see is really the so-called resting state networks. And you can get other interesting behavior for structural networks and other types of networks. But if you apply this, you can now ascribe greater detail to the function of brain regions in your network. So here, what you're seeing is the brain divided up into putative communities, and you can ask, for instance, how different nodes participate in this network and how they're related to one another in the context of these specialized communities and modules. One thing that you can look at is, for instance, the within module degree. So what is the total number of connections that a node has to other nodes in the same module? Or you could look at the so-called participation coefficient, which is a measure of how evenly distributed a node's links are. What you're seeing here is an example of applying this, where you can have a node that has lots and lots of connections, and before you knew about the communities in the network, you would say that this node is very important. It is, but many of its connections all of its connections in this case, are to other nodes in the same community. So to other nodes that potentially share a similar function. Whereas a node that's shown here in this purple community has also many connections, but its connections are evenly distributed to other nodes that are in other communities. In other words, this node here on the left, which we might call a quote-unquote connector hub, it potentially performs a more integrative function than the node that's shown on the right here. I keep losing my cursor. That we would term a provincial hub. And so looking at your degrees and your participation coefficients really gives you access to this very rich kind of functional cartography of your network, where you can divide the brain into hubs and peripheral nodes, but then within the hubs you can ask, for instance, are really these brain regions participating in very specialized functions, or are they more integrative in nature? And there are lots and lots of other statistics that you can apply once you have a community partition for your network. So that's just a very brief overview of some of the things you might want to calculate about your connectomes that you're generating in your work. What I really want to talk about now that is very important and sometimes overlooked is how you actually interpret these statistics and how do you make scientific inferences about what you're seeing. So, as I said, many of these network measures are relative. So if you find, for instance, that you have an efficiency of 0.5 in this network that you've reconstructed, how do you know that this is special, that this is interesting, that this is something that you should write about? Because many of the properties that we talked about will depend on simple features, such as the number of nodes, the mean degree in the network, or the density of the network. So how do you rule out these more trivial factors and say this is really due to the higher order topology of the network? Many of the topological differences that people are seeing could be masked, or they could be overemphasized by these trivial differences. So what you really want to say is that the property that you're finding is statistically unexpected, and that it's biologically meaningful in some way. And the way that we're going to do this is we're going to construct null models and null distributions for features of interest. So here you found that your network has an efficiency of, say, 0.6. I'm showing, I think 0.6, yeah. And you want to know whether this is statistically unexpected. A null model in this case, as we all know, is really a process by which we, in this particular case, we're systematically disrupting the topology, and we're retaining other features or properties that we think are less important to our feature of interest, such as our efficiency. So what we're doing really is coming up with a process to build up a null distribution for our feature of interest, our efficiency, and we're then benchmarking this feature of interest against this null distribution. And there are really two ways to do this. So this is, you're building up a null distribution. So this is not that, hopefully this is not that mysterious to you guys if you've, calculated a correlation or any kind of t-test before in your work. But there are really two ways to do this. One is to use generative models where you actually specify rules about how a network is formed. So you have a small set of parameters that govern network formation. So this would be a little bit more analogous to the parametric statistics that we use that often have a generative process underlying them. The other set of ways that people do this is to use what I'm calling rewiring models. And these are more akin to non-parametric statistics, where we start with our empirical network and we randomize it under some constraints, and then we keep recalculating our feature of interest to come up with a null distribution. And I'd say that these are much more commonly used in the field. So that's what I'll show you. With rewiring models, you're starting with your original network. You're going to say, I'm going to fix some properties and I'm going to randomize others. And the way that I'm going to do this is I'm going to start rewiring connections with some constraints. By far, the most common method that people use to do this is, in neuroscience, we call this Maslow-Sneppen degree-preserving rewiring, although because we all keep citing this paper by Maslow and Sneppen, although the method has been around for much longer. The way that it works is we take our network, we pick two edges at random, and we randomly swap them or rewire them. So what this does is it creates a new network that has the same number of nodes, it has the same total number of edges, so therefore its density is the same, and the degree of each node is preserved. So the node here had 4 connections before, it had a degree of 4, it still has a degree of 4 afterwards. This node had a degree of 1 before, it still has a degree of 1 after. So it embodies the null hypothesis that the feature of interest that I'm seeing is due to these low-level properties and not due to the overall topology of the network. The cool thing about these null models is that they're very flexible. So they can allow you to implement lots and lots of different scientific questions if you additionally control for other factors. So you can control for the geometry of the network, or the so-called wiring cost, directionality, and so on. And it allows you really to test a range of hypotheses. And that's what I'm showing here. What you're seeing is an original kind of network that we are starting with. This is a null model where I'm just preserving the density. So this is this so-called Erdős-Rényi random graph. I'm just placing edges at random until I achieve my desired density. This is Maslow-Sneppen rewiring, where I'm now randomly flipping edges. So I'm preserving density and degree. And you can see that there's now a lot more structure in this network. You can see a lot of prominent horizontal and vertical streaks, because nodes that were previously hubs are still going to be hubs in this network. And this is a null model where I'm actually randomly flipping edges, but I'm only doing so as long as the total wiring cost of the network is preserved. So I'm additionally fixing geometry, and what you're seeing is that now I have a very, very conservative null model that looks very similar to my original null. So these null models, there's no one perfect null model. Really, they're kind of a spectrum of increasingly stringent or liberal null models. The one thing that I just want to point out as you're embarking on this again is that these null models are so fundamental to what we do that they've actually been subsumed into many of the network measures that we routinely see in the field. So for instance, the so-called small world property, which is a ratio of clustering and path length, is actually not the ratio of clustering and path length. It's the ratio of the clustering coefficient that's been normalized by the clustering coefficient that we see in randomized networks, and path length that's been normalized by the path length that we see in randomized networks. The so-called rich club property, which we didn't really talk about, but again, I'm sure many of you have encountered in the literature before, this idea that there might exist greater than expected connectivity between the hubs of the brain, is actually defined as the density of connectivity among the high degree nodes relative to the density that we expect in a population of rewired networks. And finally, the modularity that we did talk about. There are many different ways to define this expected connection density within a putative community, but by far the most popular one is the so-called configuration model, or this kind of rewiring, Maslow-Snepen rewiring null. One thing that I want to point out is that while these are good null models to use for structural networks, sometimes you have to be careful if you want to do this for functional networks. And what do I mean by that? With functional networks, we're really talking about patterns of correlations or covariances that arise from some underlying dynamical system. And correlations by their nature are transitive. That means that if I know the correlation between node A and node B, and I know the correlation between node B and node C, I can place a limit as to what the correlation between A and C is going to be. So what that means is that if you go in and you apply this kind of degree preserving rewiring, you could generate randomized networks that never could have naturally arisen as correlation networks. You know, here you have a very strong correlation, here you have a very strong correlation, but here you have a very weak correlation. So these networks are definitely no longer positive semi-definite. Sorry, did you have a question, Fred?
Yeah, we do have a question from Francesca Castaldo. Does the parcellation template influence the structural connectivity and therefore the network analysis?
I mean, to an extent, yes. Obviously, that's a great question. I mean, it's a very common question. Obviously, there are a few papers that have shown that you can get different types of for the different features that we can calculate about connectomes. We do get different magnitudes, but at the same time, what we see is that when you compare them to the appropriate population of rewired nulls, you get the same results typically across different parcellation schemes, parcellation resolutions, and so on. I would argue that it's very good practice when you're doing this kind of work to repeat your analyses for different parcellations and potentially different parcellation resolutions. So just so you convince yourself and your readers that what you're seeing is not really idiosyncratic to the parcellation scheme that you've chosen.
Thank you.
So these rewired nulls. So like I said, when you apply Maslow-Snepen rewiring to functional networks, correlation-based networks, you can get very unnatural looking nulls. And so what you might do instead is, have you lost my slides again? Are they moving?
We're still on the choose to.
Oh, there we go. That worked. Okay. Yeah. So so what you might want to do is reconstruct your nulls in some other way. And there are lots of methods that have been around for a very long time that allow you to reconstruct null functional connectivity matrices, but not by rewiring connections, but before in the step before as you're making the actual correlation-based network. So one family of models are what are called phase randomized surrogates. What you're doing here is you're taking the original time series for your nodes. You're oftentimes doing a Fourier transform, so you're taking them from the time domain into the frequency domain, where you can jitter the phase coefficients and then bring the signals back into the time domain. This creates new time series that have the exact same power spectrum as before, but the temporal dependencies in the time series have been completely destroyed. So you can now recalculate your correlation matrices and use those as your nulls. The other way to do this is to use what are called autoregressive surrogates. So to use simple, AR1, AR2 type models to reconstruct your functional connectivity matrices. And there's this really nice paper from Raphael Lejoie who actually showed that these multivariate autoregressive surrogates can actually give you very good, very similar behavior to what you get with your original data. So they're very good null models. I just want to point out one other kind of tweak to null model testing that is, I think, becoming very prevalent in the field, and that is to incorporate information about space and geometry. We know in both structural networks from diffusion imaging and also from track tracing and also in functional networks that the probability of there being a connection between two neural elements will decrease monotonically with the space between them. So if you have two brain areas that are very far away, they're much less likely to be connected than areas that are very close together. And if there is such a connection between two brain areas, it's going to be smaller in magnitude the further they are away from one another. And you typically get these characteristically kind of exponential decreases in when you compare distance and weight. So what that means is that the constraints that are imposed by spatial embedding, which are very real because the brain has to fit between your ears, so it has finite metabolic resources, finite material resources. So it has there exists a prevalence of short-range connections, and we can incorporate this factor into our null models. We can either use generative models that have penalties for distance, or we can, as I showed you with the rewiring nulls, we can introduce additional constraints when we do our rewiring. But this is just to show you again that if you have surrogates that just take into account these spatial constraints, you can come up with null networks that look very similar and retain many of the high-level properties that that your empirical networks have. So it's something that is important to keep in mind. It's one of these things. I mean, I always think of it like if you're doing research into neurodegenerative disease or neurodevelopmental disease, and somebody asks you, are you accounting for age? Well, it depends on whether I think of age as a covariate or not, because a lot of what I'm looking at is biologically very meaningful and changes with age as well. And age and neurodegenerative disease are not completely inseparable. So in the same way, connectivity and topology and geometry are not completely separable in the brain, and it's something to keep in mind when you're studying your networks. And the last thing I just want to show you is that it's often very good to check the assumptions that you have. Many of the measures that we talked about will entail very strong assumptions about how the network works. This is one example. It's a classic paper from Steve Borgatti, who said, well, here's a network. Tell me what's the most important node in this network. And the thing here is that, well, if I think that communication takes place by nodes simply passing messages to their nearest neighbor, then I might look for the node that has the highest degree. And in that case, this is the most important node in the network. If, on the other hand, I think that messages are being passed sequentially, following shortest paths, then I might compute either the closeness centrality or the betweenness centrality of a node, in which case these two nodes are the most important nodes in the network. And alternatively, I may think that information is actually passed simultaneously, forwarded simultaneously to multiple neighbors at the same time, and then they forward that message simultaneously to other neighbors, all in parallel. And in that case, I might look for the node that has the highest eigenvector centrality, and that's the node here. So something as simple as saying what's the most important node in my network strongly depends on how I define communication or message passing in my network. The other thing is, or a very good example of this, is looking at the communities or clusters that you find. So all of us typically look for communities that are assortative, that we define communities as groups of nodes that have dense connectivity with one another, more so than with other groups of nodes. So this is true whether you're doing any kind of community detection, clustering, ICA, whatever. But there exists, and if you can certainly apply that kind of model, and you'll get yourself a set of nice looking communities. That's not the only way to define communities. And in fact, in network science, there exist a myriad of ways in which you can define what are called disassortative communities, for instance. So groups of nodes that are defined as a community based on their connectivity to some other target group of nodes. And it's a perfectly legitimate way to look at this, or to define groups of nodes that have dense connectivity to multiple communities at the same time. You're not going to detect these types of communities, disassortative communities, for instance. You're not going to detect them if you apply out-of-the-box community detection algorithms that assume assortative communities. And if you look for communities of this latter kind, like disassortative communities, you might get a completely different picture of how the brain is organized. So what you get as your final networks of interest strongly depends on what you tell the algorithm to look for.
Bratislav, we just have a question from Matthew. I think it was regarding right before for your spatial models. So it's about, can averaging across range of thresholds provide a more robust network analysis? And as a second question, how are anti-correlation typically handled in network analysis?
Great questions. So averaging across a range of thresholds, you certainly can do that. I think it'll be much more useful. I mean, first of all, it depends on whether you need to be thresholding or not. If you have, just one network, you don't necessarily need to be thresholding. But if you're doing any kind of case control comparison, then you can. I think you can also, so I guess your idea there is to find a feature of interest, calculate its magnitude across a range of thresholds, and then average it. I'd much rather see, two curves being shown where the x-axis is the actual threshold, and I would like to see consistent differences between your two populations across a range of thresholds, as opposed to summarizing it with just one kind of average measure. And then the other question here is, how are anti-correlations typically handled in network analysis? So anti-correlations you get if you're doing functional connectivity. So what this means is that you have negative values. You typically get negative values if you've done global signal regression. So the simplest way to save yourself a headache is not to do global signal regression, but I'm not advocating for one way or another. But if you have gone with global signal regression and you have negatives in your work, you have a few different ways open to you. One is what I've seen people do is to just set the negative correlations to 0. The other way is to look at the absolute magnitude of the edge weights in your network, so to get rid of the negatives. The third that I like quite a bit is that for many network measures that we have, there do exist signed analogs. What I mean by that is that, and I'll just see if I can quickly go back one slide, and there is a good example of this in my slides. So it's being shown here. You see here, this is actually a solution that I derived in a paper from a postdoc. Here, the blues here are actually anti-correlations. And there is a way to do community detection where you concentrate the positives into your communities and you push the negatives out. So for many network features that I talked about, there does exist a signed analog. And it does depend a little bit on the feature and what it's trying to capture about the biology, whether you're going to, how you're going to treat the negatives. Okay, so as the very last slide here is that, what I often see in papers is people rushing off to do finding the most complex network measure that they can, and then reporting that as their feature of interest. And the reality is that very small systematic changes in local properties could potentially manifest as more complex global effects. So this is just a very kind of simple example. You have your two groups. You found that they're a clinical population and control population. You find that they're different in terms of their efficiency. This is all very exciting, and you want to say that the disease that you're looking at has disrupted the efficiency of the clinical group. But if you can go back one step and you can look at, for instance, something like the histogram of all the edge weights in your network. And what you can see here is that for the most part, the two networks are actually very similar to one another. There just exists this very small proportion of connections that are actually different. And if you go back one more step, you actually look at the adjacency matrices that are leading to this, you see that they're actually very similar to one another. Really, the biggest effects are concentrated in this very small number of connections. So while it is true that the efficiency is smaller in group A compared to group B, really the reason this is happening is that this very small number of pathways is somehow attenuated in magnitude in group A. So I think that it makes for a much more complete and much more compelling scientific report if you actually show me where the effect is coming from, as opposed to just showing the final statistic that abstracts away all the biological detail. And I think it's important to verify why the measures that you're seeing, why they're changing, where your effects are coming from, and oftentimes to actually justify why you need a complex measure when you could have potentially used a simpler measure. And I think that in terms of the workflow for most studies, what would be useful is if your topological analyses that did some kind of graph theoretic approach are accompanied by a statistical analysis that allows you to pinpoint the origin of your effects. And there are lots of different ways that you can do this. This is one example of a statistical technique that was actually explicitly developed to help you with this, called network-based statistics, or NBS, from Andrew Zaleski and colleagues. Just one final kind of to show you, if you want to do this kind of thing in your own work, you're lucky, because there exist lots and lots of different toolboxes and different programming languages that have already implemented most of these measures, and also the null models that we use for testing. Many of them work from the command line. Many of them are more kind of with a GUI, kind of point and click. So there's something for everyone. If you're interested in reading more about this, I would highly recommend these two texts on the left that are more specific to neuroscience, one by Olive Sporns, the other one by Alex Fernito, Andrew Zaleski, and Ed Bullmore. If you're interested in network science and from a more broad perspective, they're the two kind of classic textbooks at this point, by Barabashi and Newman. And if you have work that falls in this domain, I would encourage you that you submit it to the journal Network Neuroscience that's been around for a couple of years now, and that accepts this kind of work. So just to summarize, I hope I've convinced you that neural organization can be fruitfully represented as a graph. And really what network science allows us is to formally study, to quantify, and to ultimately articulate how the connectome is organized. That these connectomes obey a small number of seemingly universal organizational principles that transcend different methodological differences. But at the same time, that representing the brain as a graph is an exercise in dimensionality reduction. We're talking about models that abstract away a lot of important biological detail, and that make a lot of assumptions. They make a lot of assumptions about how the brain works, and it's important to really verify these assumptions and to think about what you're doing. So thank you very much for your attention. I'm happy to take any questions.
Thank you so much for the talk. That was so interesting, as always. So we're slightly over time, so we'll have time for one question. So if someone, well, I see one in the chat. So Venkatesh is asking, wonder how does the hierarchical nested clusters view of the network tie into a dynamical systems view of the brain?
So that's really a question about, there is a very rich field of people who take brain graphs and then superimpose some dynamical system on top. And those dynamical systems can range from very, very simple kind of linear time shift invariant systems to more complex neural mass models and neural fields. The simple answer is that it's an ongoing question, because what you can do and what people are actively studying is actually, partitioning the brain into a hierarchy of clusters and then seeing how that influences the emergence of different types of dynamical behaviors. With graph theory, you can obviously do that. You can find a way to partition your network, and then once you have a dynamical system running on top, you can further analyze the tendency for patterns of correlations in the system to follow the underlying kind of cluster structure. So I don't think that there's any kind of agreed upon final answer as to how they're related, but it's something that can be readily answered with the methods that you saw here.
Thanks a lot.
Thanks.
So we'll stop here for the questions. That said, if Bratislav, you can still stay for the student discussions, then students are invited to come and join. So it's going to be more of an informal question answer type of thing. So of course, everyone is welcome to stay. But thank you everyone for coming today for the CIC lecture, and then we'll see you next week.